So the topic for today is Krebs cycle. So the Krebs cycle is also called as tricarboxylic acid cycle, as the first stable product of the Krebs cycle is citrate, which is also a tricarboxylic acid. Hence, it is also called as tricarboxylic acid cycle. So now let's study the Krebs cycle. So the first step of the Krebs cycle is formation of citrate from oxaloacetate. So in this reaction, the oxaloacetate reacts with acetyl CoA and water molecule to form citrate, and the enzyme involved here is citrate synthase. Okay, then from citrate undergoes dehydration, that is removal of water, and forms cis echinoctate. Then cis echinocase again undergoes rehydration, that is it takes the water molecule again, and there is formation of isocitrate. And the enzyme which is involved in this both reaction, that is from citrate to cis echinoctate and from cis echinoctate to isocitrate, the enzyme here involved is echinotase. Okay, so and then the next reaction is formation of oxaloacetate from isocitrate. So isocitrate undergoes dehydrogenation or oxidation, that is removal of hydrogen. So the hydrogen molecule is removed from the isocitrate molecule. So if the hydrogen molecule is removed, so there will be someone to accept the hydrogen molecule. Here in this case, the NAD accepts the hydrogen atom and forms NADH2. So NAD is getting converted into NADH2, and the enzyme involved here is isocitrate dehydrogenase. Since the name of the substrate is isocitrate and the reaction it catalyzes is dehydrogenase, hence the name of the enzyme is dehydrogenase. Then the next step is formation of alpha ketoglutarate. So oxalosuccinate undergoes decarboxylation, that is there is a removal of carbon molecule in form of carbon dioxide. So the name of the enzyme is oxalosuccinate decarboxylase. So the name of the substrate is oxalosuccinate, and the reaction it catalyzes is removal of carbon dioxide, that is decarboxylation, that is, so the name of the enzyme is decarboxylase. So the name of the enzyme is oxalosuccinate decarboxylase. Okay, and there is formation of alpha ketoglutarate. So there is formation of alpha ketoglutarate from oxalosuccinate by removal of a carbon molecule in form of carbon dioxide, and the enzyme involved is oxalosuccinate decarboxylase. Then from alpha ketoglutarate there is formation of succinyl CoA. So in this reaction there is dehydrogenation as well as decarboxylation. That means there is removal of hydrogen as well as carbon dioxide. Okay, so since there is a removal of hydrogen molecule, so there will be someone to accept this hydrogen atom or the hydrogen. So NAD also act as a hydrogen acceptor, and one NAD is getting converted into NADH2, and carbon dioxide is released. Molecule is released in form of carbon dioxide. So the name of the enzyme is alpha ketoglutarate dehydrogenase, since it catalyzes the reaction of that is dehydrogenation, that is removal of hydrogen. Then so this is how the alpha ketoglutarate is formed. It is converted into succinyl CoA. Then from succinyl CoA is converted into succinate. So when succinyl CoA is getting converted to succinate, so there is a rehydration, that is there is the addition of water molecule, and during this process there is a energy release. There is a release of energy, and that energy which is released, it is utilized in conversion of one GDP molecule into GTP. so there is formation of guanosine triphosphate. Since guanosine triphosphate is unstable compound, it is immediately converted into adenosine triphosphate, that is ATP. Since guanosine triphosphate is unstable, it is immediately converted into adenosine triphosphate, that is ATP. So from succinyl CoA to succinate, there is formation of 1 ATP, and the enzyme which is involved in this reaction is succinate thiokinase. Okay, so the name of the enzyme is succinate thiokinase. Then from succinate to fumarate, then the next reaction is succinate to fumarate. So conversion of succinate to fumarate, there is also removal of an hydrogen molecule, that is there is in this step also there is a removal of hydrogen atom. So if the hydrogen atom is being removed, there will be someone to accept it, and here the hydrogen acceptor is FAD. So one FAD molecule is getting converted into FADH2. So succinate is getting converted into fumarate. So the enzyme involved here is succinate dehydrogenase, as there is removal of hydrogen atoms, so it is hence the name succinate dehydrogenase. Then from fumarate, okay, from succinate we have studied till succinate to fumarate. Now from fumarate to malate. Now let's see how malate is formed from fumarate. So again there is a addition of water molecule. And fumarate is getting converted into malate, and the enzyme here involved is fumarase. Enzyme which is involved here is fumarase. Then from malate to oxaloacetate, again the conversion of oxaloacetate from malate, there is again removal of an hydrogen atom. So if there is removal of hydrogen atom, there will be someone to